Hello everyone, and welcome to today's webinar, Investigating the Effect of Diabetes on Bone Quality via Changes in Gene Expression of Glycation and Bone Remodeling Markers in Osteocytes. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GIBCO by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Rachana Vaidya, PhD candidate, Department of Bioengineering at the University of Massachusetts Dartmouth. Rachana, you may now begin your presentation.
Hello everybody, my name is Rachana Vaidya. I'm a PhD candidate in the Department of Bioengineering at University of Massachusetts Dartmouth. My advisor is Dr. Lamia Kareem, and we work in a bone biomechanics lab where we are trying to understand diabetes-associated skeletal fragility in these patients. Today I'm going to talk about how diabetes affect bone quality and how we can check the effect of diabetes on bone quality by checking the gene expression markers in bone cells called osteocytes. To give you a background about diabetes, let's look at the big problem. So, diabetes is the 7th leading cause of death, and according to the International Diabetes Federation, as of 2019, there are 463 million people in the world with diabetes. And these are the number of adults from the age 20 to 79, which is the working population. And this number is expected to increase by 51% in the next 25 years to 700 million. So we need to join hands and try to address this issue at a global scale, but also at an individual and community scale as well, if we have to stop this from reaching 700 million. So what is diabetes? Diabetes is a metabolic disease in which the body's inability to produce any or enough insulin causes elevated levels of glucose in the blood. There are two major types of diabetes. Type 1, in which the body cannot produce insulin, and type 2, where the insulin is produced but used inefficiently. Type 1 generally develops in the childhood, and type 2 is most common in the age of 40. But if you look around and you have friends and family members, most of them in their 30s are now entering pre-diabetic stage, which means it is an increasing problem of younger population. Type 1 diabetes is caused when the immune system starts attacking your insulin or insulin producing cells or beta cells. And type 2 is most commonly caused because of overweight, poor diet, and inactivity. Blame our pandemic lifestyle, shall we? So this year we celebrate the 100th year of insulin discovery. Dr. Frederick Banting and his colleagues received the Nobel Prize in 1921 for discovery of insulin. And they sold the patent for just $1 so that the whole world could reap the benefits out of it. So it's 2021, and have we cured type 1 diabetes? No, but we have come close to a cure. We now have artificial pancreas, which can monitor your blood glucose level as closely as it is in healthy patients. And that we all can say is a bridge to the cure. So let's look at diabetes complications, or what can high levels of glucose or hyperglycemia do to our body. So it can increase your chances of stroke, cataract or glaucoma, tooth decay or periodontitis, atherosclerosis or other heart disorders, nerve damage, chronic kidney disorders or nephropathy. But in the last 20 years or so, people have identified an underappreciated risk, which is increased fracture risk in diabetes. To understand the increased fracture risk, we should look at what's there in our bone. So let's zoom into our bone. When I started my PhD, I thought that, okay, this is a skeleton and there are several bones, and how do we study how the fracture risk is increased in bone? Because I thought that bone is just probably minerals. So you do probably some mechanical test and figure out what's happening on it. But I was wrong. Bone is an extremely complicated structure and beautiful with beautiful architecture. Let's look at it. At the whole bone level, the bone is divided into a proximal and distal end. At the tissue level, it is divided into two types of tissues. This whole structure is the compact or cortical bone, and there is an inner structure called spongy or trabecular bone. Now, the cortical bone has a fundamental structural unit called osteon. If you look at this microscopic view, each osteon has a central canal through which the blood vessels are running, and it is surrounded by concentric lines of collagen fibrils. Okay, so these at the microstructural level, it is made up of lamellae, which are basically collagen fibrils arranging itself into parallel sheets, which then run in concentric circles around the central canal. Each collagen fiber is made up of singular collagen fibrils. Each collagen fibril is made up of collagen molecules interspersed with hydroxyapatite crystals or calcium phosphate crystals. And so it's organic matrix plus the mineral that makes up our bone. So this is a picture of the bone of human tibia that we generally work with in our lab. We cut these human tibias into small pieces and then look at the cortical structure, and then you can see the spongy trabecular structure. The central canal is where your bone marrow generally is, but these are cadaver bones, so you won't see anything here. Bone is also made up of bone cells. Well, so there are four major types of bone cells: osteogenic cells or stem cells, osteoblast, which forms the bone matrix and makes new bone, osteoclast, which resorbs or removes the old damaged bone, and osteocytes, which acts like a team leader and maintains the bone tissue by telling the osteoblast and osteoclast of when and what to do. So what determines bone strength? Bone strength is determined by bone mineral density. This is a DEXA scan of a patient. It tells you the bone mineral density in terms of grams per centimeter square, and it gives you two scores, T scores and Z scores. T score is how good your bone is compared to a 30-year-old young adult, 30-year-old young adult bone, and Z score is how good your bone is compared to age-matched individual. So if your bone density is if your T-score is between +1 to -1, you have good bone density, nothing to worry about. If you have anything between -1 to -2.5, you have low bone density, maybe start taking calcium pills. If you have anything lower than -2.5, you are at a high risk of osteoporosis. So next time you want to check your bone mineral density, you can look at the scores and tell yourself what your problem is. Bone strength is also determined by bone architecture, or how thick these the trabecular thickness, the how big the circles are, the cortical porosity basically, and other subtle features of architecture. Bone strength is also determined by quality, and bone quality is determined by a few things. First is microdamage accumulation. If you look at this long crack within your bones, so every day when we are running, walking, falling down, or whatever, small micro cracks are being accumulated in our bone, and your bone is actively working really hard to fix these micro cracks by making new bone. How is it done? It's done by a process called bone turnover. So bone turnover is when, imagine you have the micro crack here. So the bone lining cells will be replaced by it activates the cells, and then these cells are replaced by osteoclast, which will then resorb or remove the old bone matrix. And then they undergo a reversal phase, which is basically just recruitment of osteoblast. The osteoblasts then come and deposit new bone, leading to new bone formation, and then they embed themselves into the matrix and become osteocytes. So this process happens daily, and it is what is maintaining our bones in its proper shape and size, and keeping us healthy. So bone quality is also determined by mineralization. By mineralization, I mean what's the quality of the mineral that is formed in your bone. And we still don't know this process accurately or entirely, but osteoblasts generally uh, transport these calcium phosphate material through intracellular vesicles, which then infiltrate and deposit the crystals within the collagen, and where they grow and change shape and transform themselves to make a mineralized collagen matrix. So it looks like a very delicate process. It also determines your very important for your bone strength. Bone quality can also be determined by matrix composition. So how good is your collagen? It can be measured in terms of crosslinks in your bone. There's a good type of crosslinks, enzymatic collagen crosslinks, which keeps your collagen healthy and gives it strength. And there is non-enzymatic collagen crosslinks, which are formed randomly and can affect or deteriorate the quality of collagen. So, quantity versus quality, who wins the race? Now, type 1 diabetes is associated with 6 times greater risk of fracture than non-diabetics, whereas type 2 diabetes patients have a 3 times greater risk of fracture than non-diabetics. Now, if we look at the bone mineral density scan of type 1 diabetes, they have low bone mineral density, which might help us predict the fracture risk in these patients. But if we look at the bone mineral density scan of type 2 diabetes, they have the same or slightly higher bone mineral density because of being overweight and high bone mass and body mass index. So we cannot look at their bone mineral density scan and predict the fracture risk in these patients. So that means we have to look at something else. So we have to look at probably bone quality. So how can we look at bone quality, or what sort of measurements do we use? We can look at the amount or accumulation of collagen crosslinks, the bone cells or the tissue turnover, how good your bones are at replacing and adding new bone, and the amount of micro damage that is accumulated and fracture repair that takes place in the bone. So in our lab, we study how collagen crosslinks, or AGEs, advanced glycation end products, affect bone cell and tissue turnover, which in turn can explain bone quality and can help us explain or predict the fracture risk in type 2 diabetes patients as early as possible. So what happens in a healthy bone versus type 2 diabetes bones? So healthy bone has everything in good amount. There's normal amount of glucose comings, healthy osteocytes, healthy osteoclast, healthy osteoblast, good collagen quality, very few AGEs, and there's good amount of adipocytes too. But in type 2 diabetes bones, the increased glucose coming from the blood can, one, decrease osteoblast differentiation. So new bone is made, less new bone is made, more bone is removed, old damaged bone is removed. There is increase in the advanced glycation end products, which deteriorate your collagen quality and strength of collagen. And then there is an increase in the bone marrow adiposity. And also there is a decrease in osteocyte density. Overall, does not look like a good scenario for your bone. Now, let's look at what advanced glycation end products are. So, in 1912, a French chemist called Louis Maillard wanted to know what makes us so attracted to our food. Is it the color? Is it the taste? Okay, it is the taste, but what gives our food a taste that makes us attracted to it? And he found out that during the process of heating the food in high temperatures, there are some compounds formed in the bone, in the food, called advanced glycation end products, and these give the nice brown color to your food and makes us attracted to our food. So what are AGs? They are basically when sugar non-enzymatically reacts with the proteins, amino acids in the food, or proteins, lipids, and nucleic acids in our body, can give rise to dicarbonyl compounds, which ultimately, over the period of months to years, can form advanced glycation end products or AGEs. Now, when the sugar initially reacts with the protein, it causes functional changes in the enzymatic activity of the proteins and increases its vulnerability to oxidative stress. And when they form dicarbonyl compounds, these are early glycation products, which starts causing oxidative damage and inflammatory reactions in the tissues that it starts accumulating. And when they turn into advanced glycation end products, it could be bad. It could cross-link structural proteins like collagen and deteriorate the quality. Just have a look at your skin. Why do we think you have wrinkles on your skin? Right? Our collagen is the most abundant protein in our body. It's what gives our skin the elasticity. So when the AGEs cross-links proteins like collagen, you have wrinkly skin. So we are not just aging, we are aging. Sorry. So as we age, the AG accumulation naturally increases naturally increases. But in people with diabetes, the diabetes complications, the people with diabetic complications, the high glucose or the excessive glucose in the blood can accelerate the accumulation of aging, thus accelerating our aging process. So what are the sources of AGs? There are endogenous AGs that are formed within our tissues due to the excess glucose in blood. And there are dietary sources such as foods processed at high temperatures, processed meat, cheese, sausages, etc. All the good-looking, good-tasting food, basically. So here are a list of AGs that are associated with diabetes complications. Pentosidine, it's one of the most widely studied AGE. Because it is fluorescent, it is cross-linking, and it is easy to measure. The other widely studied AG is enapsalon carboxymethyl lysine, because it is found to be in high levels in the serum of diabetic patients. And it's a non-cross-linking, non-fluorescent AG. Methyl glyoxyl is one of the early glycation compounds. So the earlier we detect the AGs, the better we can stop them. We can stop them from forming advanced glycation end products. And glucosapan is also gaining recent attention because it's found to be in higher levels than other AGEs, but it's not so studied so widely. So the recognitions of AGEs has just started to gain attention in the society, and more people would definitely, more people should work on it, definitely, as it is in our food, in our tissues, and it is causing a lot of problems. So this is a recent paper by Dr. Nakamo M. and colleagues, where they found the level of pentosidine and CML in serum of patients with diabetes, with dyslipidemia, and hypertension. So white bars are patient without diabetes, and gray bars are patient with diabetes. So you can see that the level of pentosidine and CML are found to be significantly higher in patients with diabetes, not so much in dyslipidemia, but also in hypertension. Now, if you look at what I'm trying to show you is that CML amount is in micrograms per ml and pentosidine is picomole per mg. So CML is found in much higher concentrations than pentosidine, and there are other AGs which might be in much higher concentrations than CML, but we only know these two right now. And he also found that the level of pentosidine and CML are associated with, not significantly associated with long bone fractures. Okay, good news, but are significantly associated with vertebral fractures and all types of fractures in general. I see a problem there. So let's see what roles do AGs play in the bone. Now, AGs have two roles to play in the bone, a physical effect and a biological effect. The physical effect is exerted by cross-linking the bone matrix or the collagen organic mineralized matrix. which can lead to reduced mechanical properties such as ultimate strain and stress. Okay, here you can see as the level of CML and pentosidine increases, the propagation toughness in the bone decreases. Now, propagation toughness is the ability of bone to resist fracture, okay, or so resist crack propagation when when the bone faces fracture. So that means that your bone is, if you have a small crack in the bone, it will propagate much faster in bones with high CML and pentosidine than in a healthy bone. Not good for you. AGs also exert a biological effect by altering the cellular activity, by increasing inflammation, which in turn alters cellular activity, which in turn affects the collagen or quality of collagen that is produced by the cells, and thus reducing fracture healing and increasing microgrammage accumulation, again, which leads to reduced mechanical properties and overall can increase your fracture risk. So in our lab, my work is focused on studying the biological effect of AGs in the bone. So let's look at osteocytes. Now, osteocytes are one of the 4 major types of bone cells, and they are very cool, very fascinating cells because of their size, their shape, and their location. So, firstly, 95% of the bone cells are osteocytes, but they're also the least studied cells. Their location, which is embedded in the mineralized matrix, makes them pretty fascinating, but also makes them difficult to study. They communicate with other cells, osteoblast, osteoclast, which means that studying them, by studying osteocytes, you can understand how other bone cells are playing a role. They are the key regulators of the bone remodeling or the bone turnover process. So they can sense local mechanical strains, they can sense hormones, and control the bone formation and resorption process by secreting 2 important proteins called sclerostin and rank ligand. So when the osteocyte produces sclerostin, sclerostin is an antagonist of the bone formation process. So more sclerostin means less bone formation. And it also produces rank ligand, which binds to its receptor on osteoclast and promotes their differentiation so that they can go and resorb bone. So by controlling the levels of sclerostin and rankle, they maintain the bone tissue by regulating how much bone is formed and how much bone is resorbed at any given time. They also play an important role in repair, such that apoptosis of osteocytes around the microdamage triggers the other bone cells to come to that locations and start the repair process. They themselves can resorb bone and increase the size of their lacunae, which is like the purple space around them, by secreting a compound called cathepsin K. So when the calcium demands in our body increases, such as during lactation, these cells will join hands with the other osteoblast and osteoclast in resorbing bone so that they release more calcium into the bloodstream and the calcium demands of the bodies are met. So really cool cells, aren't they? So why do we study osteocytes? Now, they are the least studied and the most interesting bone cells. Previous studies have used osteoblasts, which are precursor of these cells, to study osteocyte response for a long time. However, they recently developed an MLOI4 cell lines, but they're still early osteocytes and do not express all osteocytic markers. Now, Dr. Paula Pajovic from Boston University has worked on developing a cell line called Osteocytes 454, which expresses high level of sclerostin, which is a late osteocyte specific markers, and shows very low level of osteoblast expression markers. Thus, it's a mature osteocytic phenotype and appropriate for measuring osteocyte markers to see the only true osteocyte response. These are some pictures that I just took from my cells, and you can see they're like macrophages actually with their cool dendritic processes. and they love communicating with other cells. So if you like leave them isolated, they would extend their dendritic processes and try to connect to other cells, because I think that's very important for them to work. They join hands together and they're very good at networking. So the effect of AGs on bone cell behavior has been studied previously using MLOI4 cells, and uh, that this is the general idea of what they found. So hyperglycemia, oxidative stress, and AGs. Most people studied AGs by just incubating ribose with bovine serum albumin and making AGs, but not the effect of specific AGs. So most of the studies found decreased in bone formation, increased cortical porosity, and decreased in overall remodeling, which then leads to more AGs to accumulate in the crosslinks. So, but there are certain things that people haven't studied, like how AGE specific response differs from hyperglycemia or oxidative stress, and etc. So that's where my work comes into place. So my overall goal is, one, to differentiate the AGE and hyperglycemic response. How do I do that? Okay, let's look at what we know. Now, diabetes give rise to hyperglycemia, which in turn will give rise to increasing level of AGEs, such as CML. Now, CML, so AGEs in general are known to act on the cell by two receptors, receptor for AGE RAGE, and there is a inbuilt um, mechanism to fight the effect of AGEs via anti-glycation receptor, or AGR1, which is just one of the many other anti-glycation receptors. Now, RAGE interacts with AGE and activates the nuclear factor kappa beta pathway, which in turn leads to increased oxidative stress and increased inflammation. So we can check that by checking the levels of TNF alpha and IL-6, which are pro-inflammatory markers. Now, the AGER receptor interacts with AGE, internalizes it, and degrades it, and then releases these small AGE peptides, which are then cleared from your, which gets out from the body through renal excretion. Hyperglycemia itself can lead to rage. Okay, my hypothesis is that hyperglycemia sets the stage for AGEs to act on the cells. So when hyperglycemia increases the reactive oxygen species within the cells by acting on mitochondrial, by increasing the mitochondrial respiration, let's say, this in turn can increase the expression of RAGE receptor in cells, which is how then the CML might be acting on it. Like too many RAGE already expressed, more CML can act on the cell. Okay, so and now we are trying to check that how the glycation markers can explain changes in bone remodeling and inflammatory markers and etc. So can we say that change in glycation receptor, okay, if there is, if these guys are engaging into the receptors with their respective receptors, does they have a co-expression of sclerostin, rank ligand, cathepsin K, or OPG, which can explain that they are the reason for the bad bone quality, and which is why there is an increased fracture risk. Okay, my third aim is to investigate whether metformin, which is known to have an anti-glycemic activity, can have beneficial effects on the bone quality by inhibiting reactive oxygen species, or by having an inhibitory effect on the expression of RAGE, let's say, to then improve, which can suggest that metformin is good for people with diabetes so that they can reduce their fracture risk. Okay. So my hypothesis is that osteocytes grown in high glucose and high CML environment will have an increase in expression of inflammation and glycation markers, contributing to a disrupted bone remodeling process. And metformin will have a beneficial effect on bone remodeling via its anti-glycemic and anti-glycation property. Okay. So aim one is to first determine how high glucose regulate the expression of these markers, and then to determine how AG such as CML regulate the same markers, and to determine if metformin does help in elevating some of these markers and is beneficiary for the bone. Okay. So what do? I take these cells, grow them at 33 degrees Celsius, which is their submiss okay, permissive growth temperature, until they are confluent, and then shift these cells to 37 degrees Celsius, which allows them to differentiate. Now I use media with high glucose. I use 22 millimolar glucose. And I use an osmotic control, which has 22 millimolar mannitol to control as a control for this treatment. Then I have media with 3 micromolar CML and a media with 25 micromolar metformin. I use this glucose concentration based on previous studies. Now, serum CML concentrations in diabetes patients was found to be 600 to 700 ng per ml, which roughly translates to 2 to 4 micromolar. So I use 3 micromolar CML concentration, and I use a plasma, I found from studies that plasma metformin concentrations in patients with diabetes is generally around 20 to 30 micromole per liter. So I found, let's see if the people who are taking anti-diabetic medications such as metformin, is that enough, or do we need more metformin to account for the fracture risk? So then, so I put these cells in 6-well plates and incubate them for 9 days, and change the media alternatively at 1357. After 9 days, we see increased sclerostin expression. So we know that these cells have now differentiated and have started expressing osteocyte-specific markers. So then we do gene expression by qPCR, and I'm studying for the markers, bone remodeling markers, sclerostil, rankle, osteoprotein, and cathepsin K. I'll explain these in the later slides. I'm looking at glycation markers, RAGE and AGR1, and inflammatory markers, TNF-alpha and interleukin-6. So just these are some of the results that have been, that I can show you today. Okay, so these are some of my preliminary results. And here I look at what happens to cells or certain gene markers in presence of high glucose versus osmotic control. Now, orange bars indicate 22 millimolar glucose, and green bars indicate osmotic control or cells with 20 millimolar mannitol. Here you can see this is fold difference, which is calculated by 2 raised to, pardon my mistake, 2 raised to minus delta delta CT method, and in mRNA expression relative to a housekeeping gene such as beta-actin. So what do we see? There is a 10-fold increase in sclerostin expression, 3-fold increase in rank ligand, 2-fold increase in OPG or osteoprotein, no change in cathepsin K, 11-fold increase in RAGE, and a slight increase in AGR1. But since I don't have my statistics done, I cannot say for clearly. So these are not conclusions, but let's consider this as observations. So if this is what is happening in the cells, what does it tell us? Now, okay, don't be scared. So we have osteocytes, and osteocytes produce sclerostin, rank ligand, and OPG. Now, sclerostin has inhibitory effect on the proliferation of mesenchymal stem cells, differentiation of stem cells into pre-osteoblast, differentiation of pre-osteoblast to osteoblast, and it increases apoptosis of osteoblast. It also increases perilacunar modeling in osteocytes. So thus, overall, it has an inhibitory effect on bone formation. Okay, so then 10-fold increase means that there is 10-fold decrease in the formation of bone compared to a healthy patient. Okay, the rank ligand and OPG ratio determines how your bone resorption is taking place. So OPG is sort of an antagonist of rank ligand. It binds to rank ligand, not allowing it to bind to pre-osteoclast so that they do not differentiate into osteoclast. Now, there is a threefold increase in rank ligand and a twofold increase in OPG, which means that it's not enough to not allow for resorption, but it kind of promotes the resorption. So there are not enough OPG to bind to all the rank ligand. So the remaining rank ligand is now going to bind to its receptor rank on osteoclast, differentiate into osteoclast, and resorb more bone. So overall, and it also increases. Okay, so this is what's happening at the level of bone remodeling, that it severely makes less bone, and there is a slight increase in the bone resorption activity such that the balance on the bone remodeling process is disturbed, severely disturbed. What happens at the glycation levels? So here we can see that hyperglycemia is increasing the expression of RAGE. RAGE is supposed to be inducing AGEs, not high glucose. So why is that so? One explanation is that possibly high glucose is increasing the oxidative stress or reactive oxygen species in the cells, which are which from other studies have found to increase the expression of RAGE. Okay, some of the high glucose, there might be some CML in it, which so they can induce in it and increase the accumulation of AG in the bone matrix, thus increasing bone quality. There is no change in anti-glycation receptors, so there is no mechanism that is activated to fight the increased expression of RAGE or when the AG start coming in. So this might be leading to increased expression of RAGE, can stimulate NFK beta pathway, increase oxidative stress and inflammation, and that also can explain the increased RAGE is then sort of this co-expression of increased sclerostin, increased rankle and OPG, which overall has an effect on the bad bone quality, which might be increasing the fracture risk. So what is the significance of our research? Firstly, this was the first study to compare the effect of high glucose and CML on the same set of gene markers in a novel osteocyte cell line, osteocyte 454. It's the first study to check the effect of AG-CML on osteocytes. It's also the first study to check the gene co-expression of glycation and bone remodeling markers and inflammation markers. So what's the implications of our study? Our results suggest that high glucose is a major player for osteocyte dysfunction in diabetes patients, severely affecting the bone remodeling process. Now, bone remodeling is an important factor in maintaining bone quality. Thus, diabetes patients may be more prone to fractures as they would accumulate more microdamage, which is not repaired properly. No effect on perilacunar modeling, like as seen by no change in cathepsin K expression, suggests that this deterioration does not involve osteocyte osteolysis or direct action of osteocytes on the bone remodeling, but via its action on effector cells such as osteoblast and osteoclast. So what do we do in future? We should first culture these cells in 3D, since the osteocyte work as a network in detecting the mechanical and non-mechanical stimulus. Then we could co-culture these cells with the effector cells to see their effect in these effector cells. Then we could also look at the RNA-seq analysis to look at pathways that are similar and different between high glucose and CML treated cells to identify the most and least affected genes. And then since these osteocytes are actively sensing mechanical signals in the bone, like when we are walking or when we are running, in which we are constantly active. So mechanical stimulation of osteocytes in culture is also, it's important to check in culture to check its effect on bone remodeling and glycation markers. And then we could also identify the role of transcription regulators in managing these pathways in osteocytes. In animal models, we could use animal models of diabetes to understand what's not just happening in bone, but how the bone muscle crosstalks takes place, or the bone fat interface takes place, to see how changes in bone markers are associated and correlated to changes in muscle or fat tissue markers. Okay, and we could use CRISPR to block the rage expression and check for the beneficial and adverse effects for development of treatment therapies. And also, very importantly, it's important to identify other AGs that might be involved. So we need some high throughput screening method for different AGs and check expression of various markers to see which AGs might have the worst effect, okay, like engaging receptors like rage. Okay, so the take-home message for you guys today is that reduce your sugar and AGEs intake. Okay, how do you do that? Limit the intake of processed food or processed sugar. Food high in AGEs are generally fried and highly processed food, red meat, fried eggs, butter, cream, cheese, margarine. Choose fresh, whole foods, low in AGEs such as fruits, vegetables, soups, and whole grains. Choose different cooking methods such as stewing, poaching, boiling, or steaming. Eat diet full of antioxidants. So if reactive oxygen species is causing all these problems, have a diet full of antioxidant-rich foods such as vitamin C or quercetin, which is found in fruits, turmeric, resveratrol found in wine, so drink more wine. Get moving. Regular exercise and active lifestyle has been shown to reduce the amount of AGs in the body. And also stay happy, because evidence suggests that happiness is definitely linked to a healthier lifestyle and vice versa. So not recently, but last year, I got this notification on Instagram that diabetes has started following me. Okay, so don't wait till this happens to you. Eat healthy and start running now. Now, I would like to thank my committee members. This is my advisor, Dr. Lamia Kareem. Other committee members, Tracy Ferreira, Paula from Boston University, who gave us the cell line, and Dr. Jafri Jia. I'd also like to thank my lab members, most importantly, Tarane. She is another PhD student who started along with me, and she's trying to understand what happens in the bone at the tissue level, while I'm trying to understand what happens to the bone at the cellular level. And we together are trying to understand the changes that are having overall in the bone to explain the fracture risk in diabetes patients. I'd like to thank our funding sources, NIH and ASBMR Rising Star Award, and also for the self-support and training support that I received from Massachusetts General Hospital's Center for Skeletal Research. This is University of Dartmouth. It's really cool looking with a brutalist architecture. It's architect Paul Rudolph. You can check it out. Okay, on one last thing. So how amazing, I can't stress enough how amazing are these osteocytes. Now, what if I tell you that we, in the near future, if we wanted to, I'm not saying we have to, but if we wanted to, we could have our own Jurassic Park. Yeah, we could have our own T-Rex. We could build our own Jurassic Parks. How's that possible? So Dr. Mary Schweizer of North Carolina University and her colleagues reported the discovery of soft tissue in a T-Rex femur found in 2007. So they found a fossil of 68 million year old T-Rex, and they just took out some sample from it, digested with collagenase, and yes, they found collagen in it. What else? They also found some cells that were embedded within this collagen mineralized, fossilized matrix now. And they stained it with DAPI. They stained it with they stained it with antibodies that are specific to, let's say, human cells, to tell us that these are not from microbes or other sources. So they stained it with human-specific antibodies. or like mammal specific antibodies, let's say. And they found interestingly that these cells are osteocytes. These cells do have DNA as seen from DAPI stain. So now we have DNA, which we didn't have when they made the movie Jurassic Park. But fiction can turn very quickly into reality, as we see from space. Like, wait, are we in space travel now? What's next? We could have Jurassic Park of our own. Whoa. So osteocytes are connecting, are the cells that connected me to our ancestors, to prehistoric times, and might be the cells that could make us travel in space and could be the link between us and aliens. Who knows? Thank you.
Thank you, Rachana, for your informative presentation. Now I would like to introduce Taylor Thornton, the Global Brand Marketing Specialist of GIBCO, to give a few more words before we move on to the Q&A session.
Hello, everyone. Thank you for attending today's webinar. I'd also like to thank Rachana, our August Cell Culture Hero, for her awesome presentation. To learn more about the GIBCO Cell Culture Heroes program, view previous Heroes webinars, or nominate a friend or colleague, please visit the GIBCO Cell Culture Heroes website at thermofisher.com/cellcultureheroes. Please also stay connected with GIBCO by following our Twitter account at GIBCO and our Instagram and Facebook account at GIBCO Cell Culture. These details can also be found in the Resources tab. Thank you, Jennifer.
Okay, I'm sorry. I think we're having a little bit of technical difficulties. Can you all hear me? Okay, perfect. Let's see. Here we go. I don't know where my audio cut out, so I'm going to start here. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, if glucose is the problem, can artificial sweeteners prove beneficial to diabetics?
Hi everybody, I hope you can hear me. So I wanted to see if like how do we solve the problem of diabetes in the world? We cannot just do research on it, but we have to solve it at an individual level and at a commercial level where we now have artificial sweeteners coming in. So I try to check if artificial sweeteners do increase the AGE's formation or not. And there have been some studies done on it, and it is found that artificial sweeteners increase the amount of AGEs too, as much as glucose, sometimes more like xylitol and sorbitol increase AGEs much more, but sucralose has less formation of AGEs. So there needs to be a lot of work done on artificial sweeteners to see if they can be beneficial to diabetes. But in terms of AGE formation and the effect of AGE formations on the bone or in the body itself, it doesn't look that beneficial or promising.
Thank you, Rachana. The next question we have, can antioxidants be used to reduce some of the harmful effects?
Great question. I think antioxidant There is a free radical theory of aging which says that we are aging because of the increase in reactive oxygen species within us. So a lot of people have tried to work at in clinical trials to see if people who generally ingest more antioxidants have less AG formation or reactive oxidant stress within them, but the results are not form our reactive oxygen species at much higher levels than like the antioxidants can have a beneficial effect on it. So even if how much of antioxidants does one have to ingest, there's not a proper study on that. So, but yes, they definitely do have beneficial effect as these the problems in the bone or in the tissues are caused by the oxidative stress itself. So definitely helpful.
Thank you, Rachna. Next question here. How can exercise help in diabetics with higher fracture risk?
So osteocytes are cells that sense mechanical signals as well, and they communicate with other cells based on these signals. So mechanical stimulation of osteocytes in vitro, like in cell lines and small animal models, have shown to be beneficial in decreasing the source. So the sclerostin expression decreases when the cells are mechanically stimulated. So yes, this might be definitely beneficial, but what kind of exercise do diabetes use is a question, because once if you're in the late stage of diabetes, people have other joint issues and tendons and other bones and soft tissues are affected as well, which decreases their ability to perform these exercises. So maybe there can be better devices created in future that uh, targets these problems specifically rather than having an overall exercise benefit. But if you don't have diabetes yet, and if you are pre-diabetic, yes, definitely working out more and exercising increases your antioxidants in your body, reduces the oxidative stress, and will definitely be beneficial.
Thank you, Rachna. Our next question is HbA1c related to age, and does it do increase or decrease in HbA1c levels affect age?
Very interesting question and very important as well, because the way they test the diabetes in people is by the level of glycated hemoglobin, which is the HbA1c levels. Now, they have done correlations between serum HbA1c and Ag levels, and there are controversial results where some suggest that they are and some suggest that they aren't. But the problem is that we haven't identified all the Ags. So we don't know which Ag levels are increased or decreased with the glycated hemoglobin. So that could be a very good predictor of diabetes and the Ag-related problems if we could find the Ags that are correlated and if those Ags are the ones that are causing more problems. I hope that answers your question.
Thank you, Rachna. Our next question is albumin and creatinine ratio, or are albumin and creatinine ratio related to AGE?
Great question. So AGs are basically like when sugar reacts with protein. So albumin or bovine serum albumin, as we test in the cells. So these are given. So all sort of albumins basically do react with glucose and increase the AG amounts. And creatinine levels, which are increased in the kidneys, can tell us a lot about AG clearance. So when I talk about the AGR1 receptor, which actually helps in removing these AGs, they just break down these AGs and then these AG peptides sort of go to renal clearance. So creatinine levels, so they study the amount of AG peptides per mg of creatinine levels in the kidneys to study how these AGs are removed in the blood. So they are associated in some ways where it can tell you about how these AGs are processed within the cells of a body and could be a good indicator of whether a particular type of AG is cleared more or not based on the correlation with creatinine levels. Same with albumin. Albumin is one of the proteins that, and there are a lot of other proteins that have more effect. But yes, albumin is definitely related to the AG levels. Uh,
All right. Thank you so much, Rachana. Looks like that's all we have time for. Do you have any final comments for our audience?
Yeah, sure. So, I mean, my mom has diabetes, and she is, let's say, she's postmenopausal, or she doesn't have osteoporosis because I made her do her bone density scan and we found out that her bones are healthy. But she was on medications like metformin, and she was switched to another medication called thiazolidine. Now, that's one of the diabetic medications. The problem with diabetes is that diabetes medications have a negative impact on the bone, especially PZDs, and they do severely affect increase bone loss in these patients. So I told him to talk to her doctor to tell her that my daughter studies this, and she told me that okay, these medicines might be a problem, and the doctor said that okay, well, all the diabetes medications are actually in general have some side effect or is bad on health. So one of my aims in study is to understand if metformin is actually beneficial for the cells or bone cells or not. There are a lot of studies which are done, and metformin seems to be safe and effective, and it does help in elevating some of the symptoms or risks in bone health. But other medicines needs to be studied in detail, and there is a lot of research that is has not taken place. So there's a lot of scope for research there. And I've realized that after working for a few years in this diabetes, it has affected me in a way that now I'm like looking at a sweet or looking at something and it's like, oh my God, it's everything is screaming diabetes to me. So don't do that. Just remember that anything in excess is bad for health. So having a good healthy lifestyle and a good diet can be is beneficial at any stage of life. The earlier you start, the better it is for you. Thank you.
All right. Thank you so much, Rachana Vaidya, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.